Assalamu alaikum everyone, welcome to the Biochem. Today our topic is the regulation of hexokinase enzyme in glycolytic pathway. So hexokinase isoenzyme of muscle and liver are affected differently hexokinase, which catalyze the entry of glucose in glycolytic pathway. These are the regulatory enzyme. There are four types of isoenzyme. These are known as hexokinase 1, hexokinase 2, hexokinase 3, and hexokinase 4. These are encoded by the four different genes. Isoenzyme means they are protein which catalyze the same reaction in the different parts of our body. So in the muscle cell or myocyte, hexokinase 1, 2, and 3 consume glucose using it for the energy production. In the liver, hexokinase 4 maintain the blood glucose homeostasis by removing or producing glucose depending on the prevailing glucose concentration. So the predominant hexokinase isoenzyme of myocyte has a high affinity for glucose. It's half saturated at about 0.1 millimolar. Because the glucose entering the myocyte from the blood produces an intracellular glucose concentration high enough to saturate hexokinase 2, the enzyme normally acts at or near its maximal rate. So here we can see that the glucose converting into the glucose 6 phosphate by the help of the enzyme hexokinase 1 and hexokinase 2. Here glucose 6 phosphate is the product which inhibit the hexokinase 1 and 2. When the glucose 6 phosphate level is high enough, it will inhibit the enzyme which catalyzes the conversion of glucose to glucose 6 phosphate. So hexokinase isoenzyme of the liver is hexokinase 4. It is also known as glucokinase because this found in the liver and these catalyze the glucose into the glucose 6-phosphate. This is also known as the glucokinase. This is different from the other hexokinase of the muscle cells or myocytes. The concentration of the glucose when it is at the 10 millimolar, it will halfly saturated. So this concentration usually higher than the concentration of the glucose in the blood. The equilibrate the glucose concentration in the cytosol and the blood. The higher the km of the hexokinase 4, it allows the direct regulation by the level of the blood glucose. So when the enzyme has a low affinity for its substrate, it will have a high km value. So when the blood glucose concentration is the high, when the in our capillary, when the glucose concentration is high as it after the rich meal or carbohydrate meal, the excess glucose is transported into the hepatocyte where the hexokinase 4 is bind to a regulatory protein. So when a person has a meal, the blood glucose concentration is rise, so the excess glucose is transported into the liver, and these hexokinase 4 then converted the glucose into glucose 6 phosphate. So because the hexokinase 4 is not saturated at 10 millimolar, so its activity continues to increase as glucose concentration raised to the 10 millimolar or more. So whenever there is the concentration of the blood glucose is high, only on that time this activity is continues to increasing. So here we can see that the protein inhibitor hexokinase 4 is a nuclear binding protein that draws the hexokinase 4 into the nucleus when the fructose 6-phosphate concentration in the liver is high, and it releases the cytosol when the glucose concentration is high. So we can see that when the capillary in the blood capillary, when the glucose concentration is high, the glucose is transported into the hepatocyte by the transporter called GLUT2. This glucose then converted into the glucose 6 phosphate when the hexokinase 4 is detached from the regulatory protein and came into the cytosol. So this is only happen when the concentration of the glucose is higher than the fructose 6 phosphate concentration. Here the glucose 6 phosphate is competitor of the fructose 6 phosphate. So when the fructose 6 phosphate the rate of the fructose 6 phosphate is increasing, this will activate the hexokinase 4 to went back into the nucleus and then bind to the regulatory protein. But when the glucose concentration is high, this hexokinase 4 are detached from the regulatory protein and came into the cytosol and proceed the reaction. So that's all for today. Thank you for watching.